Good evening, everyone. Please confirm if I am audible. Just let me know in the chat box. So, myself, I am Dr. Violet, and today we will be discussing the questions, basically the Part C questions from Unit 4E, that is Immunology. So, before I start, can you all please confirm? Good evening, Malini. Loud, clear? Okay. Okay, thank you, Malini, for confirmation. So, let's get started without wasting much time. So, before I start with the questions, I also wanted to throw some light about the Josh batch that will be starting on 1st of July. So any of your friends, if you all are interested in joining this batch, please do join because it will be a comprehensive, complete overall preparation for CSIR at December 2024 as well as for June 2025. And you all will be able to access the chat support. Okay, so along with the regular classes, exams, you have options to enroll for internship, which is free. You all can access the mind maps, self-study booster packages there, study materials will be provided, AI, and now the most important is about the questions. So you'll have AI powered AIMNet e-test series as well for this Josh batch that is coming up. ToughNet e-question bank will be provided, concept table, concept wheel, concept cloud. So as you all can see, it is a full package. So I would suggest if you all want overall preparation without wasting much time, you all can join the Josh batch or you all can inform any of your friends who all are interested. And for more information, do call at the toll-free number 1800-1200-1818 or 080-5099-7000. Right? So now the first question that we are going to discuss. Good evening, students. Good evening, Vijay Lakshmi. Good evening to all of you. Okay, so let's start with the first question. It's a Part C question. If you see the question, it is related to cytokines, and cytokines is one of the most important topic from immunology, and they are vast, so you need to know the function of each and every cytokines. So let us see this question. It says CD4 plus T cells are co-cultured with macrophages in the presence of immobilized anti-CD3 antibody under 4 different conditions. A. Interleukin 4 plus anti-IFN gamma antibody. B. IL-12 and anti-IL-4 antibody. C. Transforming growth factor beta, that is TGF beta, and D. TGF beta and IL-6. So for 3 rounds to induce T helper cell differentiation identifiable by the cytokines they express predominantly. So these are the 4 conditions. Okay. Now the question is, which of the following is most likely combination of predominant cytokine expression in these cultures? Can you all try to solve this question? Do you all know how to solve this question? If you all are trying, please let me know. It's a tricky question, we can say that, because it is not very simple. Okay, you have to, there's a figure that you have to remember to solve this question. Let me see if you all remember. Okay, so for this question, what you all have to do is, you have to know under different conditions what are the various cytokines that are released. Okay, so these are the different conditions. So the answer for this question is actually option 1. Okay, now how will you get to that answer? You need to know this complete figure that I'm showing you over here. So what does the figure show? It shows that the naive CD4 plus T cell, okay, so it forms different T cells, right? That is Th1, Th2. Okay, so these are CD4, means these are T helper cells. Now again, they're of different types. Major one, Th1, Th2, T follicular, that is TFH, Th17, T regulatory. So they will form different cells under different conditions. Okay, so if you see the question, so what is the condition over here? First condition, IL-4 plus anti-IFN gamma. So IL-4, and whenever you see anti-IFN gamma, means IFN is not there. Okay, there is an antibody against IFN gamma, so IFN will not be there. Okay, so that is the question. Means you have only IL-4. So if it is only IL-4, so you can see, okay, under IL-4, Th2 will form, and what the cytokine, which cytokine is being released? That is the question, okay, in the option. So if you see, if only IL-4 is there, so it will release IL-4, 5, 13, and 10. Clear? So what we get? The first, under this condition, which cytokine is released? It is IL-4, right? Now B is IL-12 and anti-IL-4 antibody. So let us see if it is IL-12 and anti-IL-4, right? So we will consider here IL-12, right? And IL-4 is not there. So under this condition, IFN gamma and TNF alpha is released. So let us see where it is. Yes. So for B, you can see other options don't have IFN gamma, right? Similarly, here also other options don't have IL-4, right? Then let us see for C, that is transforming growth factor beta, TGF beta. So only TGF beta we have to see. So it will be this option, TGF beta, and it will give rise to T regulatory cells that will release IL-10 that inhibits immune responses. So all that functions are also mentioned. So for C, it will be IL-10. Now for D, it is TGF beta along with IL-6. So if you see this figure over here, TGF beta along with IL-6. So the cytokine that is released is IL-17 by Th17 cells. So this is the correct option, right? Did you all get this? How to solve this question? Important. Okay. Okay. So don't miss this figure. Okay. Along now, sometimes what they can do, they can write the function. Okay. And they might not write the cytokine. You might have to find the functions of the cytokines under different conditions, what all they can do. So that is why this figure is important. Go through the figure carefully and try to memorize each and everything that is mentioned over here. Now, next is question number 2. The relation between cellular immune response generated against hepatitis C virus is the critical determinant of the outcome of infection. Given below are the representative figures of cellular immune response in column 1. Okay, so these are the response in the form of graph and the various outcome of infection. Okay, so if you see the question, it is simple, match the following. Okay, and not very complicated, just 3 things you have to match. Okay, so see the figure carefully, see the graph, and match column 1 with column 2 and let me know your answers. Are you all able to solve? Try, it's simple because everything is there, you just have to match. So the curve with circles, okay, it is showing viral load, and the curve with X's mark, it is showing T cell response. Okay, and you can see the graph. On y-axis, it is the response, okay, that is viral load or T cell response, both they are showing, and on the x-axis, it is plotted with respect to time. And there are what it is, whether it is acute infection, resolution, chronic infection, so that you have to match and tell me. Okay, I see one answer coming up. You have to try again, Vijay Lakshmi. Okay, let us see, okay, how to solve this question. See, whenever we talk about acute infection, acute means it is not in the later stages of any infection, it is in the early stages. So here we are talking about hepatitis, infection with the virus. So in the early stages, definitely virus will be there, so viral load will increase, but at the same time the T cell will also respond because it is virus, right? Viral infection, so T cells, they respond, okay, especially the cytotoxic T cells, TC cells, right? So now here, normally we have to see the answer over here, it is option 2. Okay, so let us see why it is option 2. I appreciate you all are trying, good. So you can see in acute conditions, the viral load will be there and then it will be decreased because the T cell response will be there. So that is why the viral load will increase initially and then it will be controlled because of this T cell response. So actually A will match with 1, right? Now coming to B, or let us talk about the chronic part. Okay, so this is acute and this is chronic. Chronic means the viral load is very high, right? Chronic infection means it cannot be treated. It is, it is for a very long duration. It has infected the person, and the viral load is high. So that time, what will happen? The T response will be there. Okay, it is not that the T cell will not respond, but the T cell response will be there. At the same time, the viral load will be so high, okay, that the T cell response will not be able to control the viral load. That is why the graph for chronic will be viral load will be higher compared to the T cell response. Even though the response is there, but it is very less and it cannot control the load, right? That is why we are calling it as chronic, right? So B will match with 3, A with 1. Now coming to resolution. What is resolution? Resolution means the infection has been controlled, right? Yes, Anupam, correct. So that is option 2 is the answer. So when it is being controlled, when any infection is being controlled, means that the response of the cells, of your immune cells, will be higher compared to the viral load. And you can see after a particular time, the viral load has been completely eliminated. So C will match with 2. So it is option 2 that is the correct answer. Clear? Yes, Vijay Lakshmi. Anupam, right. So let's move ahead. Okay, so here you can see I have shown you a little more graphs. So it will help you all to answer the questions related to graphs. So for T cell response in acute infection, you can see what is the graph. T cell response will be higher compared to the viral load. Okay. For chronic infection, the T cell response will also be there. Okay, but it won't be able to control the viral load. Viral load is also high. So it's negatively regulated, right? So during chronic viral infection, T cell response are also generated and they can persist over time. But these virus specific T cells generated during chronic infection, they are subjected to negative regulation. So they will lose their polyfunctionality and they'll become antigen dependent rather than developing the ability to persist long term via antigen independent cell renewal. Okay, so they will not be able to control the viral load, right? And then I also wanted to show you all the B cell response. If in case, if they ask you about the B cell response, so these will be the graph. Okay, so this is the viral load for acute, and then you'll have the antibody levels that will be higher than the viral load. Okay, long-lived plasma cells will be there, memory B cells will be generated, but the short-lived plasma cells, their levels will go down after some time, right? Similarly, the chronic infection, if you see, so only the serum antibody, even though if it is there, but still the viral load will be very high. Yeah. Let's move on to the third question. Again, it's a match the following type of questions. So whenever you see in CSIR question paper, okay, match the following type of questions, I would suggest try to solve those questions. Okay, those are simple, and you have, even if you don't know all the 5 options, at least 1 or 2 you can match, and you can get to the right answer. Okay, you can conclude the right answer. So there are various subclasses of antibodies found in body fluids and body secretions. Many different functions may be attributed to these subclasses. So given below in column 1 is major functions of different subclasses, and column 2 consists of the name of the subclass. So you can see column 1 and column 2. So try to match it and tell me what is the right answer over here. It's simple. I expect everybody to answer it correctly. Very good, Anupam. Correct. So Anupam has given the right answer over here. So the right answer for this question, that is question number 3, it is option 4. Right? Yes, Malini, you are right. So let us see the column 1 and column 2. Let us match. Yes, Vijay Lakshmi, correct. So binds to macrophages by FC. So which is the antibody that binds to FC? It is the IgG. Okay. IgE can also bind. It's not that it cannot bind, but it is mentioned macrophages. So that is why it will be IgG. If they mention mast cells, it will be IgE. Okay. So A will match with 4. Bind to mast cells and basophils. So this we already now spoke about it. That is IgE will bind to it because it is a allergic antibody that will give rise to allergic reactions. So B will match with 3. C, it is the first B cell receptor. Always remember the first antibody, okay, is the B cell receptor that is generated on B cell. So it is IgM. C will match with 5. D, no major specific function known other than antigen binding. So it is IgD. Okay, I think here there is some error in typing. So no major specific function known. It is D will match with 2. Okay, it should be IgD. And then protector of mucous membrane. So mucous membrane protector is IgA because you see it in various secretions like in saliva, in tears, in the mucus, in the gastrointestinal tract, and the respiratory tract, right? So that is why the right option is option number 4. Clear? So know the functions of each and every antibody. It is very important. Even try to find out about their. So this time, if they have asked about functions, they can also ask you match the following related to their structure as well. Okay, because all the antibodies they also differ in the structure, right? So here you can see the various functions of different antibodies is mentioned. So remember about the other functions as well, okay, which can activate the complement protein. So it is IgM. Okay, IgG can also activate, but IgM is a better complement activator. Remember that. So that is another important function you need to know. And another one is which one process the placenta? It is the IgG antibody. Clear? Question number 4, it is from MHC. Now MHC is another, after cytokines, you will see questions are often asked from MHC as well. Okay. So major histocompatibility complex molecules are encoded by a cluster of genes called MHC locus. There are several reasons why an MHC molecule on the surface of a cell is important. Which one of the following reasons is incorrect? First, to display self class 1 to demonstrate that the cell is normal and healthy. 2, to display foreign peptide in class 1 to show that the cell is infected and to engage with T helper cells. Third, to display a self-peptide in class 1 and 2 to test developing T cells for autoreactivity. Fourth, to display a self-peptide in class 1 and 2 to maintain tolerance to self-proteins. So you have to find the incorrect statement over here. I'll give you all some time. Try to solve and tell me what should be the right answer. Vijay Lakshmi, try again. Let's see. You have to find the incorrect one. So the first statement says to display self class 1 to demonstrate the cell is normal and healthy. So that is a correct function, right? The MHC, in viral infected cells, they will express some other different MHC, means the cell is not healthy, right? So that is why it displays the MHC to demonstrate that the cell is normal and healthy. So that is the correct option actually. It's very, very significant, the incorrect one. No, you can easily find it out. Just need to you just need to find the function of class 1. Okay, I think Anupam has answered for this question. Yes, Anupam is correct. For question number 4, the right answer is option 2. Okay, some said 3, some said 4, but the right answer is option 2. Yes, Anupam, yes. Sometimes experimental based questions are also asked. So we will see, okay, upcoming experimental based questions. And these are all CSIR questions. So you can see it's not that every time they'll ask the same type. Sometimes match the following, sometimes experimental, sometimes these statement based questions. So you should know how to solve each and every question. Sometimes, as I showed in the very beginning, okay, it's a tricky question, not very simple. Okay, and usually students miss to remember all the detailed conditions and different cytokines that are released. So why this is option 2? Because you can see it's written. So first one I already explained you why it's correct. Second statement, to display foreign peptide in class 1 to show that the cell is infected and to engage with T helper cells. Do MHC class 1 engage with T helper cells? No, right? They engage with TC cells. So it is very significant, no, incorrect statement. You can easily find it out. Okay. So always remember, T helper cells will identify the antigen on MHC class 2. And MHC class 2 is present on APCs, antigen presenting cells. What are those APCs? Macrophages, B cells, and dendritic cells. So this is very basic thing that you'll have to remember. And TC cells, they will identify the antigen that is present on MHC class 1. Where MHC class 1 will be present? On all nucleated cells. Right? Okay, Vijayalakshmi. Now third is to display a self-peptide in class 1 and class 2 to test developing T cells for autoreactivity. So this is also correct because when development occurs, the T cells are exposed to different antigens on different MHC. Okay, so the ones that can bind to the MHC, so those will be selected, and the ones that bind too tightly, they will be removed. So that is what it means over here. So that time they are exposed to different MHC for autoreactivity. And the fourth one also, it says to maintain tolerance. So this is same thing. Autoreactivity means it should tolerate your own self proteins. Okay, it should not bind too tightly. So the ones that bind too tightly, they will be removed. Yes, Vijayalakshmi, right. Yeah, we missed that part, right? But it is very significant. Okay, so that is how you have to solve this statement based question. And this is the, whatever I said, you can see in the figure, it is beautifully shown. So this is a CD8 T cell. T cell receptor, it has CD8, it will engage with MHC class 1. So usually it is present on virus infected or mutated cells. Okay, so those mutated cells, they'll start to express more of MHC class. Okay, and CD4 is present on APCs, means they will identify the antigen that is present on MHC class 2 that is present on APCs, professional APCs. Those are the three types that I told you. Let's move ahead and see the next question. Yes, so here you can see it's a kind of a experimental question, and there are three substances A, B, and C. Given below is the pattern of immunological responses in rabbits. When first condition A is administered along with C, second B is administered along with C, third A is conjugated with B and administered along with C. Right? So which of the following? So you have to find what is A, what is B, and what is C. Okay? And by seeing this graph, so in all the different conditions, okay, the graph is showing the response of A, B, and C. So based on this response, you have to identify what is A, what is B, and what is C. It's an average question. I won't say it is very easy or very difficult. Okay, if you have understood what is a hapten, what is adjuvant, what is a carrier protein, you can solve this question. And it is not very simple also because you have to identify the responses. Are you all able to identify what is A, B, and C? See first the first and third. Okay, so if you see the first and third, you can actually get the correct answer. Uh, Nandu, need to try again. Anybody else try? Okay, yes, Anupam. The answer is option 2. Anupam is correct. Okay, so let us see how to solve this question. See, when A is administered along with C, so A along with C, so the first condition. Yes, Vijay Lakshmi and another student. Yes, correct. A, whoever has written the name as A, that is correct. So here, when A is administered along with C, so we see against A there is no immune response. Even against C there is no immune response. Okay, means A is not a protein. Okay, and C is also not a protein, we can say, because against them we are not seeing any immune response. So what is A and what is C? Let us see the third one. When A is conjugated with B and administered along with C, so that time we see response against A also and response against B also. Means when A is given only A is there, there is no response. Means A is a hapten. Hapten are small molecular weight proteins, okay, that cannot give immune response on their own. But when you conjugate them with a carrier protein, then you will see the immune response. So that is what we are seeing over here. We are seeing immune response against A and also against B. Means B is a carrier protein. Then what is C? C can be a adjuvant, because here also we don't see the response in first and third, we don't see the response against C. Let us see the second one. It is said B is administered along with C. So here also when B is given along with C, against B there is immune response. Means B is a protein, but C is an adjuvant. Clear? Okay. So the right answer is option 2. A is hapten, B is protein, C is adjuvant. Adjuvants only enhance the immune response. Okay, they cannot give rise to immune response, they cannot initiate immune response, but they can enhance the immune response. Clear? So let's move towards the next question. Okay, so here you can see, so this is a hapten carrier conjugate. So when it is immunized in rabbit. So remember antibody, so when the hapten is conjugated with carrier, antibodies will be generated against hapten also, antibodies against carrier also, and antibodies will be generated against the conjugate of both hapten and carrier also. Okay, and these are the different haptens example, like DNP, protein carrier is BSA over here in this example. Okay. So you will see antibodies against all these three types, but major type will be against the hapten, okay, and the minor ones will be against the protein, that is the carrier, and against the conjugate. Now coming to the sixth question, again it's like a experimental question. So it's related to the receptors that HIV uses during infection. Certain chemokines are known to suppress HIV infection, whereas pro-inflammatory cytokines, they are known to enhance infection. In order to explain these findings, control and chemokine receptor knockout animals were treated with pro-inflammatory cytokines followed by HIV administration, and then infection was assessed periodically. Which are the graphical representation given below best explain the experimental results? So for this, you should have prior knowledge about chemokines. Okay, so I'll just give you all the information if you all have forgotten or you have not gone through. Whenever HIV infects, okay, it needs chemokine receptor. First thing, the major receptor it needs is CD4, so that is there. But along with that, it needs co-receptor, that is the chemokine receptors, okay, where the chemokines also bind to the cells. So that is why it needs chemokine receptors like CXCR4 is there, CCR5 is there. So these are certain chemokine receptors. According to question, one is the control and one is chemokine receptor knockout animals. Okay. So they were treated with pro-inflammatory cytokines followed by HIV administration. So can you all tell what will be the correct graph over here according to viral load? Pro-inflammatory cytokines lead to HIV infection. Okay. So now just see the graph. On the y-axis is the viral load in blood and it is plotted with respect to time. One is the one graph, okay, that is for control, and the other one, the triangle one is for chemokine receptor knockout animals. So what do you all think? What will happen with respect to the viral load? It should increase, decrease in chemokine receptor knockout animals. No. Anupam, Malini, try again. If the receptor is knocked out, how will the viral load be higher? It should not be option 1. The chemokine receptor is not there. How will the HIV go and bind? Even if you add the pro-inflammatory cytokine, the HIV has to go and bind, right? Pro-inflammatory cytokines causes more infection. Okay, but here the HIV has to go and bind somewhere, but that receptor is only not there. So even if you add the cytokines, the viral load will not increase, right? Mm, no, Arzu, try again. Yes, Malini, no, that's correct. The answer will be option 3. Okay. So you can see this is HIV where it binds. It needs CD4 along with the co-receptors, that is CCR5 and CXCR4. Okay, so if there is mutation in chemokine receptor, so chemokine receptor genes, they are associated with protection against HIV infections and will delay the progression to AIDS. Okay, so whenever blocking of chemokine receptors occurs, it will interrupt HIV infection in vitro, and this acts as a therapeutic strategy for controlling HIV infection. Okay, so that is why the right answer is option 4. So the viral load, when it is chemokine receptor knockout animals, it will be less compared to the control. Okay, got it, everybody? Yes, Anupam, right. No receptor, no binding. Yes, correct. Yes, Vijayalakshmi, it is option 3. So let's move ahead. Okay. This is related to, again, you have to have the information about the cytokines. And here, let us see what is the question. So there are 3 individuals. So again, you can see it is experimental question graph, based on this experiment. Graphs are there. You have to interpret from the graph. So 3 individuals, A, B, and C are diseased. Okay, there are 3 individuals, A, B, C. Blood samples are drawn from the 3 patients as well as from a healthy control. So control is also there. An antibody level in serum and T cell cytokine response following anti-CD3 stimulation are analyzed. So you can see antibody levels for IgG, this is for IgE, and then for cytokine like IFN gamma and IL4 are analyzed in these 3 patients as well as the control. So the following is found based on additional tests performed. The doctor concludes that one patient is suffering from toxoplasmosis that is caused by an intracellular protozoa, and one patient is suffering from filariasis that is caused by a parasitic worm. So from the data provided above, identify which patient is suffering from toxoplasmosis and which patient is suffering from filariasis. Can you all try this and tell me? Somebody answered. Then have you answered for this question? Your name is not showing up. Is it for this question or for the previous one? See carefully, students. Okay. If you all want, I can help you all by giving you a little information that for protozoan infection, okay, whenever protozoan infection occurs, remember the inflammatory cytokine like IFN will increase, okay, as well as IgG will also increase. If it is a parasitic infection, means IgE will increase and the Th2 cytokine, IL4 will increase. Okay, so I have given you all some information. So using this information, can you all try to find out Who is suffering with toxoplasmosis and who is suffering with filariasis? Okay, so I will try to solve this question for you all. I don't see any answer coming up. Are you all trying? See, the right answer for this question, that is question number 7, is option A, P and Q. Okay, so what is P? P is saying patient C is suffering from toxoplasmosis. Okay, so I have mentioned here also. So patient C is suffering from toxoplasmosis, means IgG and IFN should be high in C. Yes, Malini. So you can see IgG is high, okay, for C, as well as IFN is high. Okay, and IgE in IL-4 is less. So that means it is suffering with toxoplasmosis, right? If TH1 is there, TH2 is not there. So that is correct. So C is suffering. So statement P is correct. Q, patient A is suffering from filariasis. How patient A is suffering from filariasis? If it is filariasis, means IgE and IL-4 should be high. Okay, so let us see IgE. So yes, it is high, and IL-4 is also high, right? And the other cytokine, that is IFN gamma, is less. Means again, this is a correct one, correct option, because if TH2 is high, TH1 will be less, right? So they, okay. So the right answer over here should be P and Q. Now what about R? Patient A is suffering from toxoplasmosis. Patient A, if he is suffering from toxoplasmosis, so IgG should be high as well as IFN should be high, right? So that is not correct, because you can see IFN is less. And B is suffering from filariasis. If B is suffering from filariasis, so that time what should happen? If B is suffering from filariasis, IgE as well as IL-4 should be high. Okay, and other cytokine should be less, but it is not like that. Everything is high. So that is completely not acceptable, right? So it does not fall into the correct option. So S is also incorrect. So P and Q will be the correct options over here. Correct statement. So the right answer is option A. Clear? Is it clear for everyone? So same thing is mentioned over here. Okay. We'll move ahead with the next question. Okay, Malini. Next question number 8. Following, sorry, following sets of Plasmodium falciparum sporozoites. So there are 3 sets, okay? One, normal sporozoites. Second, sporozoites with mutation in the C terminal of circumsporozoite antigen. Third, sporozoites with mutation in the N terminal of circumsporozoite antigen. They are injected into 2 groups of mice, one normal, that is group A, and one group B, where localized knockdown of heparin sulfate receptor for CS antigen in liver cells is achieved by injecting specific shRNA expressing lentiviral particles in the liver prior to infection. 15 days post infection, parasitemia is measured by counting infected RBC through GIMSA staining. Which of the following groups will show maximum level of parasitemia? It is actually a question related to malaria. Okay, if you have not read in the QB, okay, go and study about malaria properly, because this is a disease. Okay, okay, like how the sporozoites infect the liver cells. Okay, so they have mentioned in QB what exactly, the which part of the antigen binds. Okay, if you know that Then only you will be able to solve this experimental question. Okay. So here, can you all try and tell me what should be the right answer? So whenever infection occurs, so I'll give you all a hint. Okay. It uses the C terminal to bind with the liver cells. So based on this information, can you all tell which of the group will show maximum level of parasitemia? Means the infection should be high. So it infects with the C terminal. Okay, so C terminal should be maintained of the antigen. C. Okay, Malini is correct. See, for this question, the right answer is option C. Okay, why it is option C? First is normal sporozoites. Okay, so group A is normal. Group B is knockdown of heparin sulfate receptor for CS antigen in liver cells. So which will show maximum level of parasitemia? So that should be group A, because group B, why it should not be group B? Because group B, the receptor is only removed. So anyways, there will be no infection because the liver cells cannot bind to the antigen. Okay, so that is why group B should not be there in your answer. Okay, so that is how we can omit A and D. Okay, now in A also, group A, let us see. As I said, the infection will occur through the C terminal. Okay, but in option 2, it is mentioned that the C terminal is mutated. Then definitely it cannot bind. Okay, in option 3, it is said the N terminal is mutated. So even if N terminal is mutated, the C terminal is still proper. Okay, that can go and bind to the receptor. Right? So that is why the right answer will be group A with set 3. Clear? Are you all getting this? Yes, Vijay Lakshmi, correct. If yes. Yes, correct, Malini. If the antigen receptor is mutated, means no infection. So we have to see for group A, because group B already the receptor is mutated. Clear? So it's a little tricky question. Okay, if you know about how the infection occurs, only then you will be able to solve, because they have not mentioned, okay, whether it uses C terminal or N terminal. In some questions, you can see they mention, okay. In some, if they have not mentioned, so you have to know this. So for that, you need to study the various diseases. Okay, so go through QB carefully. Okay, Malini. So let's move ahead with the next question. So whatever I said is written over here. The sporozoites, they're long, slender cells covered by 45 kilodalton protein. So that is a circumsporozoite antigen. Okay. So the binding site is a conserved region in the carboxyl terminal. So that is known as region 2, which has a high degree of sequence homology with cell adhesion molecules. Okay. And then the reason why this answer is correct. Question number 9. An antigen was injected into a mouse. Macrophages and antigen prime T helper cells were isolated from this mouse to perform the following in vitro experiments. A. Macrophages were treated with antigen for an hour and then incubated with T helper cells. B. Macrophages were treated with paraformaldehyde first and then treated with antigen for an hour. These macrophages were then incubated with T helper cells. C. Macrophages were treated with paraformaldehyde first, then treated with the digested antigen for an hour. These macrophages were then incubated with T helper cells. D. Macrophages were treated with the antigen for an hour and then treated with paraformaldehyde. So these macrophages were then incubated with T helper cells. So which of the above experiments would lead to T helper cells proliferation? So this is simple question. Recently it has been asked. So for this question, we study one experiment, right? Cell fixation experiments. So it is based on that. Can you quickly solve this? No, Malini, that's not correct. So you have to see in which of the above experiments T helper cells will proliferate. Okay, for the T cells to proliferate, the antigen should be processed and presented. Okay, so that is one requirement. Correct. Vijayalakshmi is right. The answer is option 3. In A, C, and D conditions only, you will see that the T helper cells will proliferate. Why? Because A says macrophages were treated with the antigen and then incubated with T helper cells. So then this is correct. Macrophages can take the antigen, process it, and they'll have MHC class 2. So then they can present it to T helper cells. So T helper cells get proliferated. B, what it says, macrophages were treated with paraformaldehyde. Now, if you treat the macrophage initially with paraformaldehyde, they'll get fixed. They cannot process the antigen. So after that, if you add the antigen, they cannot process it. So T helper cells will not proliferate. So B is not correct. C, it says macrophages were treated with paraformaldehyde first, but now instead of antigen, you're adding the cleaved antigen. Means the antigen is already processed and then you're adding. So in that case, it can go and bind to the macrophage. Okay, so then the T helper cells will get activated. So C is correct. D, macrophages were treated with the antigen for an hour. Okay, so they'll process the antigen and they'll present it. After that, if you treat with paraformaldehyde, then no issue because it has already presented it, so it can go and bind to the T helper cells. So D is also correct. So only A, C, D will be the right answer. Okay, Anupam, it's not 2, it is option 3. So this experiment I was talking about, okay, the question is from this experiment in QB. Okay, so you can see initially if you fix it, T cells will not get activated. If you process the antigen first and then fix it, T helper cells get activated. If you fix it first and add the processed antigen, then also the T helper cells will get activated. Clear? Next question is question number 10. It's a simple question again related to how the immunoglobulins are treated and give rise to different end products. So immunoglobulins have therapeutic applications in cancer treatment, infection clearance, and targeted drug delivery. For this reason, immunoglobulins are briefly cleaved by the enzyme pepsin. Following are some of the statements regarding the brief digestion of immunoglobulin by pepsin. 1. FAB whole twice fragment is generated which retains the antigen binding activity. 2 FAB fragment having antigen binding activity and the FC fragment are generated. 3. This fragment generated on incubation with a proper antigen forms a visible precipitate. 4. The fragment generator is incapable of forming a visible precipitate on incubation with the proper antigen. So you have to tell which are the correct statements. So you have to know with pepsin what are the end products. Then only you can be able to solve the question. So again, you can see this is from QB. Okay, so there is a figure in QB that beautifully explains what happens when pepsin is added, what happens when papain is added, what happens when mercaptoethanol is added. So based on that, you have to solve this question. Can you recall what happens when pepsin is added? Is it FAB generated or FAB prime whole twice? Only FAB or FAB whole twice? So if you know that, you can solve it. Okay, Anupam, yes, that is correct. It will be option 2. Means when pepsin is added, it will give rise to FAB prime whole twice. Okay, so 1 is the correct one. So if 1 is correct, 2 is not correct. And then if FAB fragment is there, means it can bind to antigen, it can form a visible precipitate. So 1 and 3 will be correct. Okay, the fourth one it says is incapable of forming visible precipitate. So that is completely reverse statement of option 3. So fourth is not correct. So the answer is option 2. So this figure I was talking about. So you can see pepsin when you treat, it gives FAB prime whole twice. So both, so it cleaves like this. If you treat with papain, it is only FAB. Okay, so it is cleaved from here. If it is mercaptoethanol, only the disulfide bonds they get reduced. Okay, right? Yes, Anupam. Sulfur bonds break. Correct. That is for mercaptoethanol. Disulfide. So cleavage will occur over here for pepsin. Clear, students? So just remember this figure. Okay, it is an important figure. Next. Okay, so this is about although all members of a species inherit the same set of isotype genes. So it is from allotype, idiotype, and isotype. These alleles encode subtle amino acid differences in structure of antibody. Such antibodies are termed as. So there are multiple alleles of a gene, right? So the antibodies that differ because of multiple alleles of a gene. So what are they called as? Isotypes, these are constant region changes. Okay, like IgM, IgG. So these are isotypes. Allotypes, they are also constant region changes. Okay, but this you will see in members of same species. So that is allotype. Why? Because of, no, these are not idiotypes. So this allotype arises because of multiple alleles of a particular gene. Okay, multiple alleles of a particular gene. So that is why, according to question, if you see, they have mentioned alleles encode subtle amino acid differences in structure. So because this is for multiple alleles, so the right answer will be option 2. Idiotypes, these are variable region changes. So this will occur when there are different antigens, because if antigen is different, the variable region will also change. Okay, the epitope, we know it is the part of the antigen that actually binds to antibody. Okay, so according to the question, they're saying these alleles encode subtle amino acid differences. So the members of the same species, their antibody is not exactly same, right? Because of multiple alleles of a particular gene, even though the gene is same. Yes, Vijay Lakshmi, option 2 is correct. So you can see this is isotypic determinant. These are constant region changes like IgG1. So the different antibody, they differ in the constant region. Allotypic determinants, even if the same antibody, if you see in the same species, but it is two different mice, belong to two different strain. So the constant region is almost same, but still there are slight differences. So this is because of multiple alleles of a particular gene. So that is allotypic determinants. Idiotypic determinants, they vary in the variable region, as you can see, against different antigens. So here mouse IgG1 against antigen A, here G1 against antigen B. Clear? Okay. So that is about this question. Now moving on to the question number 12. So this will be the last question and then we can stop. Which one of the following is not true for the alternative pathway of complement activation? First, alternative pathway uses the same membrane attack complex as classical pathway. Second, alternative pathway does not require antigen antibody interactions. Third, alternative pathway produces C3 by the same route as the classical pathway. Fourth, certain microbial surfaces have physicochemical properties that may result in activation of alternative pathway. So, which one is not true? So, usually from complement system, statement based questions are asked. Okay, they don't have much of experimental questions. Can you all tell which is not true, students? Okay, I see one student answering, others. So, Anupam, according to you, which is the option? Okay. What about other students? C3 in alternative already made. C3 is for classical. C3 is used from classical pathway. Okay. See, C3 is already there in the body. Okay. So that is why for this question, the statement that is not true, it is option 3, because alternative pathway does not produce C3. C3 is already there. The complement protein is there in the body. Okay. So alternative pathway produces C3 by the same route as classical pathway. This is incorrect. Okay. Even in classical pathway, C3 is not produced. It is already there. Only thing is it is being cleaved and further processing occurs. Yes, very correct. It is option 3. So option 1 is alternative pathway uses the same membrane attack complex as classical pathway. This is correct. Okay. C5, B6, 7, 8, 9. So all this forms the MAC complex, right? So that is same for both alternative and classical pathway. Second, alternative pathway does not require antigen antibody interaction. That is also correct. Only the classical pathway needs antigen antibody interactions. Alternative pathway just needs the microbes. Okay, it can bind to it. Then third one we already discussed. Fourth, certain microbial surfaces have physicochemical properties that may result in activation of alternative pathway. So that is correct. So the microbes have like LPS, okay, whatever is their constant. So they can be identified by the alternative pathway proteins, okay, the complement proteins. So certain microbial surfaces have physicochemical properties that may result in activation of alternative pathway. So whenever they see the pathogen, so this C3 will get activated. Okay, so that is how alternative pathway starts. Yes, A, correct. So the right answer is option 3 over here. So you have to remember this, flow chart, the flow chart. Okay, it is very important from complement system. If you know this, you can easily solve all the question that is asked from complement system. So you can see this is the classical pathway, this is the lectin pathway, and this is the alternative pathway that starts from C3. Okay, so C3 can identify the pathogen and then they get cleaved and further steps occur, ultimately leading to the MAC complex. That is same for all three, classical, lectin as well as alternative. Clear? So that's all for today. So just share the information about Josh batch that I already explained you all about, and share it with your friends, and whoever needs it, they can join the batch that will be starting from 1st of July. Thank you everyone. So I'll leave with this thought. I can accept failure. Everyone fails at something, but I cannot accept not trying, by Michael Jordan. Thank you. Okay, Vijayalakshmi, thank you for the sweet comment. Definitely these interactions that we have through the question answer series, it helps you all to know which part you all are, you all need to revise, where you all are lacking. Okay, so this really helps the students. Thank you for the encouraging statements. Welcome, welcome. Okay, thank you everyone.